everyone, welcome to the channel. In this video, we are going to discuss the first chapter of biology, that is fundamental unit of life. As we already know that all living forms are composed of microscopic units called cell, right? So it is a basic structural and functional unit of all life forms. It was first observed by Robert Hooke in dead cork slice in 1665. He described this in his book Micrography. Cell is the term is derived from Greek word cellula, which means small room. First living cell was discovered by Leeuwenhoek. Protoplasm inside the cell was coined by Purkinje in 1839, and it was discovered by Felix Dujardin and named as sarcode. Okay, so now let's see some statements of three different scientists and formulation of cell theory out of it. So these are all three scientists, Clayton, Swan, and Virchow. In 1838, Clayton stated all plants are made up of cell. Right? In 1839, Swan says all animals are made up of cell. In 1855, Virchow said that all cells comes from pre-existing cells. So considering these statements, cell theory was formulated. That says all living beings are composed of cells. Cell is the basic structural and functional unit of life, and all cells arise from pre-existing cell. But viruses are exception to cell theory. Is this clear up to this? Now let's see different shapes of cells. Okay, so cells are of variable shapes and sizes, which is according to their function. Generally, they are spherical, but they may be elongated like tracheids, branched like nerve cell, round like biconcave, round like this mesophyll cells, and columnar like epithelial cells, amoeboid like white blood cells, right? So these are all different shapes of cell, different shapes of different cells, right? Let's see what is the size of cell. So size varies depending upon the type of organism, correct? Some are microscopic and some are visible with naked eyes. Okay, they range from 0.2 μm to 18 centimeter, and smallest cell is mycoplasm. Okay, and the largest isolated single cell is egg of an ostrich, and the longest cell in our human body is nerve cell. So now let's discuss about the different cell organelles. Before that, let's see the types of cells. Okay, so on the basis of number of cell, we have two different type. One is unicellular, and other one is multicellular. What do you mean by unicellular? So organisms that have only one cell, that is unicellular. Examples are bacteria, amoeba, paramecium, etc. Okay, then we have multicellular organisms that have many cells and differentiated body parts, that is multicellular. Okay, there is division of labor there in multicellular organism, like in human and trees, plants, correct? So next type we have on the basis of organelles and type of organization, correct? One is prokaryote and other one is eukaryotes. So prokaryotes are primitive organism, which have incomplete cell. They are not organized. Okay, they do not have nuclear membrane. And there is only one chromosome present, and the nucleus is less developed without clear nuclear membrane, right? And in eukaryotes, the cells are well developed, more than one chromosome present, and advanced nucleus with nuclear membrane and nucleolus present. Okay, next we have let's discuss about cell organelles. So eukaryotes have relatively larger cell with a nucleus and membrane bound specialized structures called cell organelles. Okay, so let's first see the cell wall. Okay, so it is the outermost covering of the plant cell. It is only present in plant cells. Okay, it is absent in animal cell. It is strong, thick, porous, and a non-living structure. Right. So it is made up of cellulose and hemicellulose. It provides definite shape and strength to the cell. So it is permeable. It permeates other outer things, correct, and allows entry of molecules of different sizes, and it has the characteristics of repair and regeneration. Okay, so this is about cell wall. Next we have cell membrane, or you can say plasma membrane, right? So this term is given by Nageli Okay, it is the limiting boundary of each cell which separates the cytoplasm from its surrounding, and it is responsible for regulating what moves in and out of the cell. Okay, it is found in both plant and animal cell, unlike cell wall, which is only found in plant cell. And this plasma membrane is made up of protein sandwiched between lipid bilayer. So this lipid bilayer, this fluid mosaic model given by Singer and Nicholson. So it is selectively permeable. This plasma membrane is selectively permeable, unlike cell wall. Okay. So this membrane is flexible. It can be folded, broken, and again reunited. Okay. So let's see some how molecules transport across the plasma membrane. Okay, as we know that plasma membrane is selectively permeable, right? So there are two different ways through which the molecules are transported from plasma across plasma membrane. First one is diffusion, and second one is osmosis. What is diffusion? In diffusion, solutes move from higher concentration to lower concentration. As you know the example of agarbatti, right? Okay. Next we have osmosis. In osmosis, solvent or water moves from higher concentration to lower concentration. It is the movement of water molecule. Okay. Then the it can also be called as the diffusion of solvent. Okay. So it is again two types. What is it? Endosmosis and exosmosis. Endosmosis means here movement of solvent across occurs into the cell. Okay. In exosmosis, movement of solvent occurs outside the cell, right? And this is actually largely depends upon the characteristics of the environment surrounding of the cell. Okay, the surrounding it is a comparison between the inner and outer surrounding of the cell. So there are three conditions there, three solutions which we can see in osmosis. The solutions are isotonic solution, hypertonic solution, and hypotonic solution. So in if a cell is in isotonic solution, what happens? The concentration of this isotonic solution is almost equal to the concentration of cytoplasm. So it doesn't really have any effect on cell. Okay, the cell remains the same, as you can see in the picture. Next we have hypertonic solution. In hypertonic solution, concentration of solution outside of the cell is more than inside. So due to this, the cell loses water. Okay, that is why it is shrinking. It is the plasmolysis happens in hypotonic solution. Correct. In next one we have a hypotonic solution. In a hypotonic solution, concentration of solution outside is lesser than the cytoplasm of cell. So due to this, the cell swells up and bursts. As you can see, the vacuole is all swelled up and the cells also swells up. Correct. Next, let's see what is the next cell organelle. This is nucleus. Okay. So, nucleus is the most important cell organs which directs and controls all its cellular activities, like brain does to our body. Okay, hence it is also called brain of the cell or headquarter of the cell. It is organized or membrane bound in eukaryotic cells only. Okay, it was discovered by Robert Brown in 1831. The nucleus is covered by double layered membrane and it has pores on it to regulate movement of materials inside the cell. Okay, it is nucleolus and majority of which is nucleus is filled with chromatin, which helps in transmission of character. Okay, and nucleolus helps in making ribosome. So this nucleus, it stores DNA, makes RNA and protein also. It controls all the metabolic activities as we already have discussed, and also regulates cell cycle, right? Next we have cytoplasm. Okay, so this cytoplasm joins the nucleus and cell membrane. Okay, it was discovered by Kolliker in 1862. Provides shape to the cell. It is jelly-like material in which organelles are implanted. Correct. It also provides the site of biosynthetic and catabolic pathways. It is divided into two parts. First is cytosol, and second, which is a aqueous soluble part, contains various fibrous protein forming cytoskeleton. Okay, and it doesn't carry any cell organelles in it. Cytosol doesn't carry any kind of cell organelle. Second one we have what is it? Cell organelles. It is a living part of cell having a definite shape, structure, and functions. Correct. So now let's see what is it. Endoplasmic reticulum. Okay, it is a cell organelle which forms network of membranes, tubular structure scattered in cytoplasm from starting from nucleus to plasma membrane. Okay, it joins literally joins the nucleus and plasma membrane. It was discovered by Potter, Claude, and Fulham. Okay, so, this endoplasmic reticulum is present in all cells except prokaryotes and mammalian erythrocytes. It transports material between various regions of cytoplasm and between nucleus and cytoplasm. It forms endoskeleton and contains some secretory proteins. Okay, it helps in the synthesis of what? Fats, steroids, cholesterol, etc. So, it is also called manufacturing unit of cell. Okay, now you know why it is called manufacturing unit. So endoplasmic reticulum is now again divided into two types according to its structure. Okay, first one is rough endoplasmic reticulum. Second one is smooth endoplasmic reticulum. Okay, RER and SER. So the rough one, it contains ribosome on its surface. So this texture is very rough, and hence is called rough endoplasmic reticulum. And it is mainly made up of cisternae and vesicles. And the ribosome on its surface help in protein synthesis. That is it for rough endoplasmic reticulum. Next, smooth endoplasmic reticulum. It has no ribosome in it, like that is in rough endoplasmic reticulum. Correct? It helps in lipid and steroid synthesis. It also helps in membrane biogenesis. Okay. Next organelles we are going to see is mitochondria. Okay. So it was first seen by Kolliker in insect in the year 1857. In 1890, Altman coined it as bioblast. And in 1838, Carl Benda renamed it as mitochondria. So this is the long history of mitochondria, the naming of mitochondria. So mitochondria is a double membrane bound structure where outer membrane has specific proteins and inner membrane is folded inside towards the matrix to form chambers called cristae. Correct. So the space between two layers is called perimitochondrial layer. It is the site of Krebs cycle or aerobic respiration. Okay, so ATP synthesis occurs in here. Okay, it happens to synthesize ATP and also stores the energy in it. Okay, so this is called the powerhouse of cell. Now you know why mitochondria is the powerhouse of cell. Okay. Next, it contains matrix, which contains enzymes for Krebs cycle, and mitochondrial membrane contain enzymes called oxysomes for ATP production. Okay. Next, we have ribosome. So ribosome is a complex of RNA and protein. So it is known as ribonucleoprotein. As it does have ribosome and also protein. Okay, so here RNA is around 62% and rest is protein. It is located inside the cytosol. It is composed of two subunits, smaller and larger. Okay, these subunits are joined to each other by interaction between rRNA of first subunit and protein of other subunit. Okay, by decoding mRNA binded to smaller subunit, I mean, and amino acid gets added to the larger subunit, and this is how the protein synthesis occurs. Correct. Next cell organelle we have is lysosome. So these are tiny sac-like structures, membrane bound vesicular structure formed by the process of packaging in Golgi apparatus. They do not have a definite shape or size. They contain hydrolyzing enzymes called acid hydrolysis. So they're like a stomach of cell as they digest foreign materials, waste material or damaged cells. And they're kind of waste disposal system. At times when a cell is damaged, they digest their own cell. Okay, so hence they are called what? Suicidal bag of cell, right? Next we have Golgi apparatus, or you can say Golgi body. Okay, so it was discovered by Camillo Golgi. It is consist of many flat disc shaped sacs or cisternae stacked parallel to each other. Okay, it is absent in prokaryotes, mammalian RBCs, and sieve cells. It helps in formation of lipids, middle lamella, melanin synthesis, and also it packages the lipids and protein which are synthesized by endoplasmic reticulum. As it packages it and transfer it, so that is why it is also called as post office of cell, right? So it is the site of glycoprotein and glycolipid synthesis also. Next we have plastid. Okay, in this plastid, this term was given by Haeckel. The chloroplast was discovered by Leeuwenhoek and coined by Skimper. Okay, so the plastid is a double membranous discoidal structure only found in plant cells, that you already know, right? So depending upon the types of pigment present in it, there are three types. That one is leucoplast, okay, which doesn't have any pigment. Next we have chromoplast, which are colored pigments other than green, and next then we have chloroplast, which are green pigments or chlorophyll present. Okay, so this is the site of what? Photosynthesis. This is the site of photosynthesis, so it is also called the kitchen of the cell. Okay, next we have centrioles. Okay, centrioles, so these are hollow tubes that give organelles their shapes. The main function of centrioles is to help chromosome move aside the cell, and it is present only in animal cells. Okay, this helps in cell division. Okay, so then we have vacuoles. Okay, it is called as the storehouse of the cell, as it stores water, excretory product, or toxic metabolic product. Okay, other inorganic and organic products are also stored in here. Okay, so they are bounded by a single membrane called a tonoplast. They form contractile and food vacuole in many organisms. In animal cell, vacuoles are smaller and numerous. There are many. While in plant cell, there is only single vacuole, which occupies 90% of the cell volume. Okay, so vacuole helps in maintaining the osmotic pressure of the cell. Okay, and it is also called as what? Storehouse of the plant. Okay, sorry, storehouse of the cells. Now let's see the difference in plant and animal cell. So first we have cell wall. Plant cell have cell wall. The cell wall is present in plant cell, and animal cell doesn't. A plastid present in plant cell and not in absent in animal cell. Vacuole, there is only one vacuole in plant cell, and there are numerous small vacuoles in animal cell. The size of vacuole in plant cell is large, and the size of vacuole in animal cell is very small. Then we have centriole, which is absent in plant and present in animal. It helps in cell division. Okay, then we have the position of nucleus. In plant cell, you can always see that the nucleus is located sidewise, and in animal cell it is located in center. The nutrition of plant cell is autotrophic and animal cell is heterotrophic. Estrogen division is present in plant cell and absent in animal cell. Cytoplasm division in plant cell occurs by the cell plate, and in animal cell it starts with the cleavage. Okay, so that's it for the difference of plant and basic difference of plant and animal cells. Okay, this is it for this chapter. Do let me know if you have any doubt. If you find my video useful, then please share it with your friends. Thank you for watching.